I'll collect feedbacks for you, but I decided not to because there is no from our group, and yeah, I think I'll give you the feedback after the talk, and it should be enough.
Okay.
I can also ask to Emre Hoca. Hello, Amir. And by the way, do you think how we proceed with the journal club is a appropriate way to get the most out of it? Do you think, I mean, isolating people who haven't studied enough is a nice way to invoke people to study more? Ayşe, sence iyi bir yöntem mi? Bu yaptığım sen, yani eğer yeteri kadar çalışırsa insanlar makaleyi o zaman alıyoruz ya. Yoksa
Evet.
almıyor. İyi diyorsun. Tamam. Biraz katı çünkü.
Yani anlayamadıkları yerler olabilir tabii ama
Hı. Tamam.
herkes
Okay.
aynı düzeyde olmadığı için.
Evet. Okay, for the guys who don't want to join the club, you can leave if you like. And Kübra, bye bye.
Okay. Thank you to all of the things.
Yeah, we are thankful that you were with us.
Bye.
Does anybody know where Emre Hoca is? Should we call him? Yes, thanks.
Hocam Emre Hoca birazdan katılacakmış. Siz başlayabilirsiniz dedi.
Ha, tamam. Okay. Today, Eylül Korkmaz will give a presentation. She's a bachelor student. And this is one of her first presentations ever. We wish good luck to her. She prepared quite heavily. And I'll record the talk. Okay, Eylül, the stage is yours.
Okay, hocam, thank you. Hello everyone. Today I'm going to present you this article named Arabidopsis halleri shows hyperbioindicator behavior for lead, and lead accumulation is spatially separated from zinc. First of all, I would like to introduce you the term hyperbioindicator. This term is firstly used for a plant called Gombrehana closeni, and this plant has unique properties about metal accumulation. For example, one of the properties is this plant contains much higher concentrations of metal in their roots. Normally, hyperaccumulator plants accumulate their metals in their shoots, in their above ground tissues. However, these plants accumulating its metals in the roots. It doesn't translocate the metal to above ground tissues. And another property is These plants, as you can see here, there is a saturation curve about metal concentrations and soil available metals. These plants shows a linear correlation in the saturation curve, which is much more similar to bioindicator species rather than hyperaccumulators. And also another property is these are much like the hyperaccumulator species. These plants also accumulate higher concentrations of metals in their bodies. So these three unique property makes a plant hyperbioindicator. Okay, I am
May
passing
I ask you a question?
Yes, of course.
So why some plants are called excluder? Do you have any idea?
I actually didn't look at the excluder term at all, but
Yeah, but you can make a guess if you like.
maybe yeah, those plants, I think they have very tightly upregulated metal exclude mechanisms. So they don't even contain the metals. They don't even take the metals, even when the soil metal concentrations are too high. They are trying to avoid that metals.
Yeah, I see. Okay, and next question. Why do you think the name is indicator? What does it indicate?
If there is a metal in the soil, those indicator plants is definitely contain that metals in their bodies.
Oh.
If that metal is potential, if you're investigating this, you can look at the plant body and understand if there is metal in the soil or not.
Oh, I see. So you had a little bit connection problem, so I just rephrase what you said.
Okay.
You said indicator means comes from the word to indicate, and then these plants indicate the soil metal concentrations. So if you don't want to, for example, measure soil metal concentration, but you can directly measure concentration in these plants, and then you understand what the concentration in the soil for this specific metal is. You see it's a linear curve for hyper bioindicator, or also for bioindicator, there's a linear line. So you just measure the metal concentration in the plant, and you can predict very well the metal concentration in the soil. That's why it's called bioindicator. Okay, very good. Thanks.
Okay.
Hojam, can I please ask a question?
Please.
Hojam, on which basis we can say that how quantity a plant can you can say that and we can say that this plant is a hyperaccumulator?
I couldn't hear the question because of the background sound. Can you repeat?
I want to ask about how much quantity of any heavy metal.
There's someone else in your room who is also connecting. If it is the case, can you turn off the microphone?
Mic on है off?
Yes. Now. Yes, much better.
So how much quantity of a heavy metal if a plant absorb, and we can say that this plant is a hyperaccumulator plant?
I think these concentrations change by the biogeographic region or the plant species, but if we look at this saturation curve, we should definitely see this hyperaccumulator curve like a concave shape. So for a plant to be hyperaccumulator, it must contain high concentrations of metal, even when the soil available metal concentrations are low. I don't know the exact concentrations because I think it's changed, but there must be a shape like this when we get the graph. Am I right?
Not really.
I read at one place that the hypercumulator plants are absorbing almost 100 to 1000 times more than the normal plants.
Yes.
Okay.
So, in fact, if you look at here, so we see a line here. So only the plants above this line are called hyper, and this line is 1000 microgram per gram dry weight. Okay, if the metal concentration exceeds this, these plants tend to be called hyperaccumulative, but not always. I mean, one of the requirements for a plant to be called hyperaccumulator is to exceed or to have such a high concentration of metal. But it's not enough. It's not sufficient.
So that concentration is specific to lead or for all other heavy metals as well?
For all the heavy metals.
Okay.
Yeah, but if you ask me why 1000 but not 900, for example, there is no very logical answer to this. I mean, people just say above 1000 is hyperaccumulating.
I understand.
Okay.
No problem.
Yeah.
Okay, should I
Thanks for the
continue?
question. Yes, please, Eylül.
The lead is one of the most widespread soil contaminants in the world. According to data of ATSDR, it is the second most dangerous metal in the soil in terms of frequency, toxicity, and potential to human exposure. And it is important to study lead in plants because, as we can see, the lead in soil, it can enter the plants and it can enter the food webs via plants. So if it enters the food web, it would be very harmful to human health. The lead as a highly toxic compound, as a highly toxic element, it can affect every organ system in the human body in a bad way, as you can see here. So it is important to study lead in plants in order to prevent this toxicity for humans and other animals. So, however, studying lead has some difficulties, has some challenges. First of all, lead's bioavailability and mobility in soil and hydroponic cultures are very low. So it is important to apply lead or lead deficiency while we are investigating lead in the plants. However, researchers cannot do this in proper way because its bioavailability and mobility is really small. It precipitates with phosphate in the soil, and it cannot reach the plant body. It cannot be taken by plant body. And as a consequence of lead, this root-to-shoot translocation factors for lead is really small. This means that when we search the leaves of the plants, we can see lead in the leaves. However, these leads do not come from the roots, do not come from the soil. It comes from the air. It comes from dust, and this also manipulates the results of the studies. So, what can we do about studying lead is, first of all, we must limit the aerial contamination in the leaves so that we can see the correct results, correct translocations of lead from soil. And also, we must use mediums with low phosphate and low pH conditions in order to prevent lead to precipitate with phosphate and in order to mobilize lead in the soil, in the medium. Okay, I'm passing to next slide if you don't have questions.
I have one question.
Yes.
Okay, you said that it precipitates with the phosphate in the soil. It make a complex with the phosphate, or do you mean that it leaches down with the phosphate? I didn't understand.
Can you repeat the last sentence of questions?
You said that it precipitates with the phosphate in the soil.
Mhm.
I want to ask that does it make a complex with the phosphate, or do you want to mean that it leaches down in the soil?
I think it reaches down because it becomes not photo available for plants. So I'm not sure about that, but I think it leaches down in the soil, as I understand.
Okay.
I'm not sure, hocam.
Good question. Thanks. I think it makes a complex.
It makes complex. I understand.
Yes.
But when it makes a complex, it still cannot enter the plant, right? Plants cannot take it.
Yes, because when it makes a complex, then it's not soluble anymore.
Okay.
I think.
So in our paper, in the experiments, the soil comes from Bestwig, Germany. This soil is really contaminated with mining and metal smelting activities. So it's a metalliferous soil, contains high concentrations of metals. They use barium chloride and hydrogen chloride to determine the soil extractable available metals to make a low pH conditions and to measure the soil metal concentrations actually. And also they use the accessions comes across to Europe. They use 12 accessions. 5 of them comes from metalliferous sites shown in red dots, and 7 of them comes from non-metalliferous sites with green dots. And in figure 1, they took this soil from Germany and the accessions across to Europe.
Sorry, from which spot did they get the soil?
Here, Bestwig, Germany. And you see that the red dots are here mostly. Metalliferous site.
Okay.
So they get the accessions on the soil, and they grow these plants for 6 weeks in those metalliferous contaminated soil. The aim of this experiment is actually to test hyperaccumulation, wheat hyperaccumulation potential under controlled laboratory conditions, because they want to exclude.
Yeah, sorry, what's metalliferous and non-metalliferous?
Metalliferous contains high concentrations of metal because it is affected by the mining or metal smelting activities on those biogeographic regions. So soil contains much more concentrations of metals compared to non-metalliferous soil.
Okay.
Okay. The main aim here is to test wheat hyperaccumulation potential under controlled laboratory conditions, because they want to exclude aerial contamination as an underlying cause of the high lead concentrations in the leaves of plants. At the end of 6 weeks, all accessions showed high concentrations of lead in their leaves. And origin of the accession actually did not affect the lead concentrations significantly. For example, the metalliferous sites contain nearly 600 micrograms of lead, and non-metalliferous sites was nearly 650 micrograms. However, these horizontal lines, the black lines, represents the reference plant Arabidopsis thaliana. Arabidopsis thaliana contained only 145 micrograms of lead in their leaves. That's why we can say that all Arabidopsis halleri accessions contained much more lead than the Arabidopsis thaliana reference plant. And also in figure 1B and C, they look for the zinc and cadmium concentrations. By contrast with the lead results, zinc and cadmium had different zinc and cadmium concentrations had affected by the origin of the accessions. In general, metalliferous accessions contain less level of metals than the non-metalliferous sites. And also in figure 1D, they look for the titanium concentration. This is actually a control for soil contamination. If there would be a contamination in the leaves from soil, we expect to see high concentrations of titanium in the leaves. However, we do not see any correlation between lead and titanium, and this confirms that there is no contamination for these results, for these experiments.
Just for clarification for those who haven't read the article carefully, I mean, these red ones are the accessions that are taken from metalliferous sites, like near the smelters, maden ocağı. And the green ones are maybe from cities or, I mean, from soil which is not containing a lot of metals. Okay. Mm-hmm.
So I'm passing to figure 3.
Yeah, and maybe a little bit more clarification. So why do you take from 2 different regions? Because I think you assume the ones from the metal side are adapted to these conditions, and the ones not from the metal sites are not adapted to conditions where excess of heavy metals exist. So they assume these lines may give different responses, and maybe in the future then they can also examine what makes them behave differently and so on. So they want to look to the big picture. That's why they choose a lot of different lines. Okay.
Okay. In figure 2, they look for the saturation curves of these elements. As I said in the beginning, for a plant to be hyperaccumulator, it should contain high concentrations of metal, even when the soil available metal are low, metal concentrations are low. And here we can see that zinc and cadmium show the classical hyperaccumulator behavior in the curves. However, leads show the linear correlation, very similar to Arabidopsis thaliana. This black dots represents the Arabidopsis thaliana plant as a control. And these two are showed a nearly linear correlation, which is much more like a bioindicator behavior. So they choose 3 of the accessions from the experiment 1. I forgot to say, Laut 3 was 7. These are showed the strongest metal accumulation in the experiment 1, in the soil experiment. And they choose also CF1. This showed the weakest metal accumulation. That's why they continue their experiments with these accessions.
I want to add something here. Very important, please everyone listen carefully. So we said if the concentration is above 1000, it's an hyperaccumulator. Now here 1000 is here, and as you can see, as the lead increases, it's almost there. The concentration is almost 1000. So it might be considered as a hyperaccumulator. But having such a high concentration is not enough. Even this was like 1200, even if it was like this, we couldn't say it's a hyperaccumulator. For a plant to be hyperaccumulator, we need to see this type of curve. So even the concentration of the heavy metal is low, the plant immediately takes a lot of metal and sends it to the chute. It's an active process for hyperaccumulators. So for hyperaccumulators, heavy metal, normally toxic heavy metal, is not something they want to get rid of. That is something they want to accumulate in their chute. Maybe because they want to deter the bugs. So the animals, when they eat them, the animals will die. So they don't prefer to eat the hyperaccumulator ones. It's like a defense mechanism. That's why even under low concentrations of heavy metal, in hyper, I think I made my point. So the curve is very important. Did you want another explanation, or do you have any questions, especially Mohammed Zubair, about these curves? Emre Hocam merhaba.
Merhaba.
Okay, if no question, yes, Eylül, please go on.
Okay. In figure 3, they already did the soil experiments and they want to further investigate the lead translocation in the hydroponic culture experiments. They are still trying to understand the ability of Arabidopsis halleri to translocate lead in the above ground tissues. They use a nutrient solution with low phosphate concentrations. As I said, wheat has a high tendency to precipitate phosphate, and they want to prevent this. And 5 micromolar lead is exposed to plants for 3 weeks. At the end of 3 weeks, we see that root growth inhibition is apparent for all of the accessions, as you can see in here and here. As you can see here, root growth inhibition was apparent. And also they are looking for other measurements like root length, root fresh weight, and shoot fresh weight. We see that all the accessions show the decreasing pattern under lead exposure. And the wall 7 accessions show the sharpest decrease in this manner. It affected the most. That's all I say in this figure.
Eylül, did you have a look at the accessions? Like they respond differently to high lead, and I wonder if there's a logic behind this. For example, under high lead, by the way, should we say lead or lead? I think lead.
I say lead. Let me.
Ben lead diye biliyorum.
Let hocam.
Lead. Yeah.
It is lead.
Sorry.
But it's okay. So here, if you look at, for example, ZIP1, let me use the annotation tools. So ZIP1 accumulated the most lead. Wall 7 accumulated the least lead. Can we make, sorry, it's not lead accumulation, it's root length. So this plant is affected from the lead most, this plant affected from the lead least. Can we develop any logic? I mean, why this one affected less, this one affected more? Because we know they are collected from different sites.
Mm-hmm.
Do you have any ideas? Did you check which one is from which? I mean, which one was from metalliferous soil?
Yes. Let me check my notes. I had noted that in somewhere, but I cannot find this in here. But I also think the same thing, but I couldn't find an answer to this. I also tried to search a bit, but I don't know. I cannot give explanation for this.
Eylül, before making the explanation, can you look at the paper, read the figure legend of 2, figure 2 legend? it tells from where they come from.
Yes, CF1 and LAV3 comes from metalliferous sites and Vol7 comes from non-metalliferous sites.
Okay, now according to this, make your conclusion.
Okay, LAV3 normally lives in a low metal concentration soil compared to other ones, and that's why it's maybe affected most when high concentrations of high toxic metal implied to that one. It's not lives in the highly metalliferous soil. Yes.
I would agree. Yeah.
Now I have one question. When you look at the control plants in the first genotype, as you can see, there is a slight difference in the root lengths. And compare it to the second genotype and third genotype. Only I am talking about the control plants. Do you think this image is representing the variability in the graph? Look at the those bars. Are they representative?
I think no. Vol7 looks root length of Vol7 says very high degrees, but it doesn't look like a higher root length, longer root.
Yeah.
Wow.
I am telling this, and I will tell you the reason why I am extremely careful about that. When I submitted my first paper to Plant Physiology, one of the critics was Marilu, and they criticized this difference in the same way I criticized this paper. And that critique was so sharp, so intense that we were almost failing to publish the paper. And I know why she criticized this, because she criticized me later on in a meeting as well, from the same point. So as you can see, this group, they are very famous in this area. Again, in their paper, the same thing happens. And most probably the reason for this is that while you are taking the image, you just randomly select plants and put them onto this blackboard. But while you are taking the calculations, the entire set, maybe you have 30 plants, 40 plants, I don't know how many, but your data in the graph is much better representation of your results. It's just the final results. But this image might not be good. And while you are taking the image, you need to be very careful, make sure that you put the same size of the plants. Otherwise, people will criticize you.
Yeah, I'm very happy I took this lesson without any damage. I mean, not trial methods, you know.
So that's why what I do, what I always tell my students is that do the experiment, take your measurements, and look at to the result of your measurements coming from different genotypes. And now you know the average size, length of the root. Then take the plant that looks to the same size, that's average mean size, and put them next to each other.
I see, I see. Good idea.
Okay, I am testing the figure 4 then. They try to further investigate the metal accumulation, and this time they're looking shoots and roots separately. Again, still what is the word? Expose 5 micromole lead to each accession. And first thing to say is all Arabidopsis halleri accessions contained 4 to 12-fold larger lead than Arabidopsis thaliana. so they contain higher concentrations of lead anyway. And when we focused on the shoot and root concentrations separately, we see that in here, shoot concentrations was nearly 600 maximum. But when we look at the roots, it exceeds thousands of micrograms of lead. We can calculate the translocation factor from here. They calculated, and we say that root to shoot translocation factor for lead is very small compared to zinc. Here are the calculated values for both elements. And also, we in the beginning we said that for a plant to be hyperaccumulator, it must accumulate their metals in their roots rather than shoots. This also confirms that property about Arabidopsis halleri. And also, it is interesting that when we focused on the zinc concentrations in here, when we expose 5 micromolar lead to plant, the shoot zinc concentrations has reduced in here. I think it's interesting.
Mm-hmm. What do you think could be the reason for that?
It is, I think, related to second claim of the article. They think that lead and zinc are spatially separated in the above ground tissues, and I think this must be a clue for them to think this at the beginning.
But can you put it under any logic? I mean, they are separated inside the plant. That's one fact. Another fact, if you take one, the other is left behind. But do you see any relation with these two facts? Or they are separate facts?
No, they are not separate, I think, but maybe lead is a high toxic compound, and plants should not accumulate lead actually in their bodies. So this is affecting the other metal concentrations negatively. This may be affecting plants, organisms negatively in this way. Is it because of this?
The logic is not clear to me, how these two facts are related to each other. I mean, is one of them explains the other, or they are more like separate? Which one explains the other?
Are you talking about the translocation and the zinc concentrations?
No, in fact, we're
Yes,
talking
again.
about something which we will see later on. You said zinc is separate from lead, but maybe we talk about it later because we haven't seen this figure yet.
Okay.
Okay, but still, can you give any explanations why zinc concentrations decrease as the lead increases? And is it really so?
Yes, in shoots, it seems like this.
Okay. In the roots?
In root, there is no difference. Actually, it increases. I think. No.
Yeah, I think they don't say, they don't come up with any explanations about it.
Yes, they don't.
Now, I have a question to everybody. When you look at this zinc concentration graphs, wall 7 is supposed to be sensitive compared to the other two, right?
No, we cannot say.
Yeah.
It's just
Now, so in the first two, SEEP and load, so basically we have seen that their root was less affected from the lead, and they accumulate more lead, meaning that most of they are like acting as hyperaccumulators. But on the other hand, the wall is coming from a normal soil originally. And here we see that it is less hyperaccumulating, or I would say sensitive, but don't think this sensitivity similar to the Arabidopsis thaliana sensitivity. So among the three, the most affected plant is the wall. But in this plant, when you look at the zinc, in the control conditions, in the shoots, let me just highlight that 1 second. Here, so it accumulates the highest amount of the zinc. But in the roots, it accumulates less compared to this more tolerant cultivar or genotype. But when you start to add the lead, of course, there is a decrease. And as you can see, the decrease in this plant is much higher. And here in the root, the zinc concentration is increasing. And when you compare the increase in between these two, this plant and this plant, the increase in the wall is much higher. How can you explain this?
But involved, right?
So there is something interesting happening in wall that actually is affecting the distribution of the zinc, especially under lead accumulation. So somehow lead and zinc are maybe competing for the same transporter in the vascular tissues, most probably. And somehow it is affecting the overall distribution of the zinc. And I think this RNA sequencing found something interesting related to this.
Did they do RNA sequencing?
They had the RNA sequencing in this paper, right?
Yes.
They're in the supplementary, but I didn't put in the here.
Okay.
I didn't understand.
I don't know why they put it in the supplementary if they did RNA sequencing.
Okay, let's
Sorry.
continue then.
Okay.
I need to clean this.
So just a second. So Eylül, these numbers here are very abstract to me. So do you really understand what they mean? So what is different according to these numbers between lead and zinc?
These are the translocation factors from root to shoot. They are looking for the shoot concentrations, and they divide them to root concentrations and get the factor, root to shoot translocation factor.
For example, what is the
as I
factor for C1? For C1. I think it's this number divided by this number. So if you look
I
at
think I have a problem with the internet connection.
sorry. We can hear you.
Okay, it's okay now.
If you look at zinc, it's 8 times more in this plant. But if you look at lead, look at this and this. It's in the shoot. It's much less. Now, I have another question for you, Eylül. Maybe a difficult one. We have read so many papers. We never saw translocation factors. Why do you think in this paper, particularly, the translocation factor is important to consider? between lead and zinc especially, but not also manganese. Mhm.
Lead, as we said, that lead is very immobile element. So in the soil, in the hydroponic system, as well as inside the plant body, maybe to understand, maybe to see clearly that this is immobile, this do not go to above ground tissues that much easily compared to other metal zinc.
But there is another reason, I think. There is a big question about lead and Arabidopsis halleri. It's also in the title of the paper. I mean, I think this puts this is an evidence to answer this question. This is answering one big question, one of the big questions of the paper. I think this is put particularly for that. That's why they especially compare zinc with lead. Do you have an idea?
About the spatially separated part?
No.
About a property of hyperbioindicators is they accumulate the metals in their roots rather than shoots. So are we let in the roots, not shoots? We
Yes.
clearly see that.
Exactly. You're on the right track. So this plant is a hyperaccumulator for zinc. And the question is whether this plant is a hyperaccumulator for lead.
Yes.
And in the previous slides, they said probably not because the concentration is low. Then they said probably not because the curve is not like this, but instead like this. And here they compare these two and they say this is less than 1. So it's also another evidence that this plant cannot be considered as hyperaccumulated. But
Yes.
mhm.
Okay. I am passing to figure 5 now.
Yes, please.
So they look at the root shoot concentrations in soil, also in hydroponic system, and here they want to direct evidence of translocation of lead to above ground tissues. So they do xylem sap analysis, and lead was detected in all Arabidopsis halleri accessions xylem sap. This is a direct evidence of translocation of lead to above ground tissues, actually. Here, the lead results are similar to those iron results, iron concentrations, but much smaller than zinc, calcium, and magnesium, they said. And that's because, in fact, calcium and magnesium is used as a control here for xylem sap analysis to show that the xylem sap volume and concentrations are in the normal range. So this concentrations of lead are correct. And I want to highlight that CF1 accessions show the highest lead concentrations in the xylem sap because they are going to use these accessions in the next figures, next experiments because of this.
That's why they chose C1 for further analysis. Okay.
I think there is no question. In figure 6, they demonstrate that lead is translocated to above ground tissues clearly with xylem sap analysis and other experiments. And now they are looking for the spatial distribution of lead in the petioles and leaves. They choose CF1 from xylem sap analysis. For the experiment, they take the cross sections of petioles and leaf main vein, and they analyze this with LAICP-MS, laser ablation inductively coupled plasma mass spectrometry. This is a analytical technique that enables us to do highly sensitive elemental analysis to directly on solid samples. So they can look for the elemental distribution by using this technique. When we look at the results, we see that in the petioles, there is no spatial difference between lead and zinc. They both allocated in the epidermis, here the edge, near edge ones, and the central vascular bundle. However, when we look at the leaves, zinc and lead allocated in different tissues. In the leaf main vein, lead was again near epidermis and central vascular bundle. You can also see this in here, colloquialization map. The red line represents lead and it increases in the epidermis and vascular bundle and epidermis again. When we focus on the zinc here, we see that zinc was allocated in parenchyma cells surrounding the main vein. Here you can see this P represents parenchyma and zinc is increasing in here. In leaf lamina, I think the separation can be seen more clearly. Here, lead is still in the epidermis of the tissue, as you can see in here, of course. And zinc stayed near the mesophyll cells inside the leaf lamina.
Mm-hmm.
Okay, is it clear?
So what was the main aim of this experiment, doing this experiment, according to you?
They are looking at the spatial distribution of lead in the above ground tissues, and they think that it's separated from zinc in the leaves and petioles.
Do you think it was surprising for them to see the lead in the middle of the leaf vein?
Maybe not, because as we can see here, no, I don't know. I just understand it falsely.
So
Are you saying leaf main vein or vascular bundle in here?
vascular bundle, yes. It's also, I mean, almost the same place. Do you think
Maybe
that vascular bundle was surprising for the others, or was it more expected?
maybe it's not so surprising, because as lead increased in the vascular bundle, zinc was also shown in here. So maybe it's a potential place to metals to accumulate in the leaf main vein.
So let's assume the others didn't do the experiment and they prepare for the experiment. So would you assume, if you were one of the experimenters, to see lead in the vascular bundles? It's the vein of the leaf and also the okay.
I want to say yes, because if they prove that it translocates to above ground tissues, it must use the veins actually. So there is no other way, I think.
Thinking, but there's even a better, more the previous figure, in fact.
A xylem cell, okay. Xylem cell is connected to veins, I think. If it's seen in the xylem cell, it must be seen in the veins also.
Yes, I mean, Yeah, you expect, I would expect it to see in the veins because it's in the dialect. We know it's in. Okay, very good.
Okay. Should I pass the next figure?
Yes, please.
Okay. Figure 7. This is hard for me. Hardest figure for me. In here, we know that is an immobile element. So it is immobile inside the plant also. So in order to mobilize the lead inside the plant, it must bind to some elements, some ligands. So in here, the aim of the experiment is to understand which elements does lead binds inside the plant, into the plant. And this results, this experiment will be meaningful if we want to investigate which tissues or where or which tissues absorbs lead mostly, or which proteins or receptors does it bind inside the organisms. If we are wondering these questions, this figure would be more meaningful. And in here, they use L3 Xanex spectra. This method uses X-ray absorptions to understand the sum of the elements and the location of the elements. And in here, they already know the curves of these acetate, phosphate, and sulfate compounds. In here, while we are saying acetate phosphate, we actually talking about the oxygen atom inside the acetate because lead is binding the oxygen atom inside this compound. In here, lead is binding the phosphorus atom, and here it's binding the sulfur atom. Okay, they already know the acetates curve and phosphates and sulfates curve and their shapes. And when they get the leads curve, this is leads curve, they are looking for the similarities between leads and the other compounds curve. The more lead is binding to an element, its curve is much more similar to those elements. For example, in here, lead's curve is very similar to acetate's one in terms of shapes. That's why the lead acetate complex is high in percentage. Lead is, we can say that lead is binding to acetate mostly to oxygen ligands. And also, previously, they did experiments about predominant binding of lead to oxygen ligands in the cell wall. This report also confirms that reported this Xanex analysis of Arabidopsis halleri also supports this concept because oxygen ligands in the cell walls are likely to be sites for lead sorption, they say.
Mhm.
That's all I can say for this figure.
I think you described it very well. So, any questions about it?
I have a question. Eylül, are you doing your master's or PhD?
I am undergrad student, but Seçkin Hoca told me this figure before.
Okay.
How many hours have you studied for the article again?
I think it exceeds 30 hours or something.
30.
Yeah.
30.
Yes, because I get the article 1 month before the presentation.
Now, before reading this article, did you also read any other articles related to this topic?
No.
No. This is the
I
first
think-
time you are reading about this topic.
Yes, I'm attending journal clubs, but I think that accumulation is the first one.
And how many presentations have you given in your life again?
0. Maybe 1 or 2 in the English class. 5-minute presentation.
Thank you very much.
Okay, you're welcome. I have a conclusion.
Yes, please show us that.
Okay. When I read the article, it was not so clear for me to understand what aspects does Arabidopsis halleri seen as a hyperbioindicator. What clues does they give us? So I want to conclude that in figure 1, they show us that Arabidopsis halleri shows stronger accumulation of lead relative to reference plant Arabidopsis thaliana. This is a classical hyperaccumulator plant behavior, we can say that. But in figure 2, they showed the saturation curve and the lead concentrations showed a nearly linear response in this manner, which is much similar to Arabidopsis thaliana. And linear correlation is a classical behavior of bioindicator plants, actually. But in figure 4, they show shoot and root concentrations separately, and we see that Arabidopsis halleri accumulate the metal lead in the roots rather than shoots. This is a property of hyperbioindicator plants. So they propose the term hyperbioindicator for Arabidopsis halleri according to these 3 evidences. And also, the second claim of the article was in the leaves, zinc and lead was allocated to different tissues. Zinc and lead was spatially separated, and we can see that in the figure 6 clearly. I think they showed us in the colocalization map. In the leaves, lead was choose to stay in epidermis tissue, whereas zinc was allocated in mesophyll cells, as you remember. That's all I can say for this article.
Did you like the article?
Yes, I like it. I think that was a good written article. I didn't understand figure 7. I didn't understand the method, but also I couldn't correlate it with the other parts of the article, other story of the article. However, when you explained to me, I get it a bit.
And let me tell you, so I think all research is done, can be classified in 2 different ways, descriptive and the story. And this one is also a bit descriptive. So if you are very new in the field, like this lead accumulation is not studied so heavily before, so you need more also descriptive figures. This describes, but it doesn't have to be in the story, part of the story. But still, they also, I think, manage very well to have story and also descriptive figures together. So that's why I liked the article very much. Also very well written, I agree. We are almost out of time.
Mm-hmm.
So let's go and come back again.
Okay. Her geçen hafta okuyan sayısı azalıyor gibi geliyor.
Evet. Maalesef. Yani insanları çok zorluyoruz tabii. Yani bu makale okumak çok zor. Benim de mesela saatlerimi alıyor yani. Hani az değil, zor bir iş. Başkasının ne biçim alacak? Herkes yapamıyor bunu. Normal yani.
Bence yapmaları gerekiyor. Yapmaları için de zorlamamız lazım.
Zorlamamız gerekiyor. Evet.
Yoksa buraya gelmeyip okumadım, oh ne hala ben girmeyeyim diye kolayına kaçacaklar bu sefer de.
Hı hı. Aynen öyle.
Bu makaledeki teknikleri Türkiye'de yapabileceğimiz bir yer var mı? görüntüleme tekniğini özellikle.
İngilizce mi anlatayım, Türkçe mi?
Fark etmez.
So they use two different techniques here, LAICP and ZANES. So
Hı hı.
in Turkey we don't have ZANES because for ZANES you need to have a huge facility like one facility like this Hadron Collider in Europe. So you know it's a very big circle, very big circle. So you need a facility like this. Only few places have such a facility. But if you book it, you can use the facility for free.
Hmm.
And for LA ICP-MS, I'm not sure if it's possible in Turkey. I mean, it might be possible, not for looking for biological samples. I would assume they use the technique to look stones, I think, like in geology, I guess. But I'm not sure. But I know one of the others very well, this Katarina Fokelvikus. She collaborates very well. She's a very nice person. If you have any important phenotype, you can talk to her about this imaging techniques.
Şu şey için micropixel analizi için.
Var mı orada? Did they use micropixel here?
Bu sanki bir yerde kullanmışlar gibi, metotta yazıyor.
Olabilir. O çizgilerin olduğu yerde kullanmışlar. O da yok. So for micropixel you again need this deep character. If I'm not wrong.
Interesting.
About the others, this Yutek Kumar is very famous, very perfectionist. We have read her paper, I think, before. And Stefan Clemens is chemistry. He works in a completely different field than most of the other people, I think. He looks specifically at what is collating what with constants and so on. Also very successful guy. Okay, any questions about the paper? Anything? Okay, if not, we can stop here.
Yes, absolutely.
And I wish to thank Eylül for this very beautiful presentation. We are amazed with the performance.
Thank you for having me.
It was a great pleasure really to watch such a presentation. Okay.
Görüşmek üzere. Eylül, tebrikler.
Teşekkür ederim hocam. Görüşürüz.
Eylül,
Hadi kendinize dikkat edin.
tekrar gir. Ben sana feedback vereyim Zoom üzerinden. Olur mu?
Tamam.
Hadi bay bay.
Bye bye. Bu slaytta bu bioindikatör nedir ve excluder nedir, kelime anlamları nereden gelmiş sorularıma doğru cevap verdim. Hyperaccumulator'ın tanımı sorusuna yanlış cevap verdim. Aslında şeyde yazıyordu makalede, discussion'da. Bir slide sonra. Bir sonraki slayta geçer misin? Şimdi burada lead is an environmental pollutant dedim. Tamam, zaten bunu anlatıyorsun, doğru. Fakat bunu daha heyecanlı hale getirmek lazım. Lead is the second most most dangerous. Neden? Çünkü bu slaytın aslında amacı ne biliyor musun? Neden benim anlattığım şey önemli? Bunu anlatmak. Ve yani lead environmental pollutant, evet, ama plastik de environmental pollutant. Başka şey ama bunun olayı yani en fazla ikinci.
Hı hı.
O yüzden o şekilde anlatsak daha doğru olur. Genel olarak başlıklara çalışman lazım. Genel şeyi gördüm. Şimdi örneklerini de vereceğim. Yeteri kadar spesifik değil, vurucu değil. Daha genel ibareler. Onları daha spesifik yapmak çok önemli. Bir sonraki slayta geçelim. Burada da başlık doğru. Net is harmful. Yani tamam, olabilir. Burada anlatırken bunu bir sürü yazı var ve işte bakın böyle diyorsun, geçiyorsun ya, onun yerine bir tane en önemlisini anlatabilirsin. Mesela brain'i anlatırsın. Yani işte böyle memory loss yapıyor, headache yapıyor, irritability yapıyor. Ama sadece brain'de değil, birçok sistemde sıkıntı yaratıyor. Ve işte çocuklara anlatacaksan, yani çocuklarda daha da tehlikeli deyip geçebilirsin mesela.
Tamam.
Çünkü bunu yapmadığınız zaman şey gibi oluyor. İşte ben buraya koydum, siz bakın gibi.
Hı hı.
Ama en önemli noktalarını vurgularsan sorun yok diğer yazıların kalmasında. Bir ya da iki ya da üç tane en önemli nokta. Evet, üçe geçebiliriz. It's difficult to study lead in plants. Burada kursunu çalışmak değil de kursunun transportunu çalışmak gibi bir şey desek. It's difficult to study lead transportation ya da lead transportation in plants. Arada çok büyük fark var. Çünkü birisi daha spesifik, dikkat edersen. Daha meaningful. Yani eğer yeteri kadar spesifik olmazsan anlam oluşmuyor zihinde fazla. Yani kolay oluşmuyor. Bizim amacımız o anlamı oluşturmak. Evet. Burada mıydı? 4'teymiş. Bir önceki slaytta tekrar döner misin? Burada şey oldu. Bu problems çıkmadan önce bayağı uzun uzun konuştun ama bembeyaz slide açıktı ya.
Evet.
O çok iyi olmadı. Onu resimlerle güçlendirsen söylediğin şeyleri, çok anlamlı olmayan resimler bile daha güzel olurdu. Muhtemelen ayrı bir slide bir öncesine öyle bir resimini koyabilirsin. Çok da mühim bir şey değil de. Ondan sonra measure kelimesini measure diye okuyorsun. Ben de öyle okuyordum. O measure okunuyor.
Beyaz süre olan değil mi?
Evet. Evet.
Neyse. Ne diye okunuyor?
Measure. E ile. Measure.
Measure. Tamam.
Bir sonraki slide. Burada main aim deyip çok güzel açıkladın slaytın hani bu deneyin amacını. Ve bunu bütün deneylerde yaptım. Yani benim takip ettiğim. Hepsinin çok güzel amaçlarını açıkladım. O sayede insanlar için çok anlaşılır oldu. Bunu işte yani ben diğer lab arkadaşlarımla da konuşuyorum ve onları da ikna etmeye çalışıyordum. Bir türlü yapamıyorlardı. Demek ki yapılabiliyormuş. Onu gördüm. Yani oluyormuş yani uğraşınca. Şaşırdım yani. Çok düzgün şekilde o nedenlerini anlatmanı, bu kadar başarılı bir şekilde anlatabilmeni. Bir sonraki slide. Bir sonraki. Bu fotoğraf iyi oldu değil mi? Fotoğrafın olması iyi oldu bence hemen atlamaktan sonra. D'yi yine güzel açıkladın. Yani başkası dediğim tabii yapılması gerekeni yapıyorsun aslında ama standartları sunumda yeteri kadar yüksek tutamadık şimdiye kadar. D'yi mesela hiç anlatmadan geçerdi insanlar. O çok güzel oldu burada, average demand falan. Burada daha sık yapabilirsin. Ne kadar vaktin alıyor bu animasyonları koymak?
Yani az vaktim oluyor.
Az vaktin alıyor. Belki bunların arttırabilirsin sayısını.
Hı hı.
Slide numaraları kayboldu bundan sonra. Slide numaralarını otomatik olarak vermenin bir yolu olması lazım. Onu istersen Google'dan bak.
Tamam.
Hı hı. Tamam. Şimdi burada önemli bir nokta söyleyeceğim bak. Bu figür gerçekten çok önemli ve sen bence çok güzel yine anlattın. Fakat yine de anlamayan bir sürü insan oldu. Nedeni şu. Bunu göstermeden önce bu deneyin şimdi amacı ne dedikten sonra 2 tane figür koysaydın. İşte hyperaccumulator olmayan bitkinin şeyi nasıl? Hyperaccumulator olanın nasıl? O ikisini koyduktan sonra şimdi işte deney yapanlar acaba şeyde nasıl olacak diye baktılar dediğin zaman, bu şekilde anlattığın zaman şu figürü insanlar bekliyor olacaklar ve çok merak ediyor olacaklar. Yani evet, acayip bir şekilde nefes almaya bırakıp hani acaba bunda nasılmış diye onu bekliyor olacaklar. O havayı yaratmak çok güzel olurdu. Bir background vermiş olursun, anladın mı? Yani sunum sırasında bir background tamam en başta veriyorsun. Fakat bazı teknikler için özellikle ve böyle çok yanlış noktalar için bir background slide versen sunumu oradan oraya taşır. Çok farklı yerlere taşıyabilir kaliteyi. Yine notları almışım. Hep slaytların eğimlerini çok güzel söylediğini not almışım. Bir tane kelime apparent kelimesini apparent diye mi okuyorsun? Apparent. Ben de çok karıştırdım. It's apparent. Ona bir baksana. Onda bir bozukluk gördüm önce. Ya şeyden emin değilim ama figürlerin caption'larını koysan daha mı iyi olur bir tarafa bilmiyorum. Hani okumak isteyen caption'ları okusun diye. Çok da emin olamıyorum.
Ben de öyle düşünüyorum. Arada kaldım. Koymadım sonra.
Aslında bunu koymuyorlar genelde. Tamam, ondan emin olamadım. Neyse. Bir tane sonra burada. Sonraki. Evet. Bu da değil. Bir sonraki. Evet. Burada higher dedim ya, herhangi bir comparison yapar yapmaz mutlaka neyle compare ettiğini oraya koyman lazım. Higher in the roots compared with Arabidopsis thaliana gibi. Hani bu bir alışkanlık olsun istersen.
Tamam.
Bir dakika, çizme anlatırken çizdin ya bazen. O gerçekten güzellik katıyor yani. Acayip iyi katıyor yani. Donuk olmuyor. Yani o iyi oldu. Geçebiliriz. Demişsin bir yerde sanırım. Burada evet. Burada direct evidence olduğunu söyledin ya bunun. O çok kritik bir noktası makalenin ve onu özel olarak üzerine basa basa vurgulaman çok iyi oldu bence. Sonra bir sonraki slaytta burada uzun uzun metodolojiden bahsettim. Bu harika oldu. Gerçekten yani insanların anlamadığı kısım metodoloji. Elbette ki sen zamanın bir kısmını buna ayırman gerekiyor. Bunu çok iyi yaptığını düşünüyorum. Ki bunu bana sormamıştın yani. LA, ICP-MS'i değil mi? Ben sana anlatmadım onu sanıyorum.
Evet, ben araştırdım. Aslında tek bildiğim o cümleydi ama tam başka bir şey sormadılar.
Öyle mi? Tamam. Yani cihazı görmediğin için ben cihazı görünce kafama oturdu gerçekten. Çok normal. Yani işten kaçmadığını da gösteriyor. Zor kısmına odaklanman gerekiyordu ve zor kısmından kaçmayıp odaklandın. Bir sonraki slide. Tamam, bu mu? Bir dakika. Yok, bu değil. Burada. Burada bir tane sıkıntı oldu. Şu. Şimdi burada önce cümleyi söyledim. Şu soldaki PB and Z'ne allocated different tissues in the leaves. Önce bunu söyleyip sonra figürü anlattın. Bizim yöntemimiz her zaman önce result'tır, sonra result'ın conclusion'ıdır. Yani önce figürü gösterip bak işte burada işte koyu renk fazla miktar, açık renk az miktar gösteriyor. Gördüğünüz gibi ortada işte koyuluk. Bu da provascular tişeye denk geliyor falan deyip ondan sonra da bunun conclusion'ı işte PB ve Z'ni gördüğünüz gibi farklı yerlerde. Sırada yani bir problem oldu sadece. Bir sonraki slide. Tamam, bunu zaten sanat eseri gibi anlattım. ve son slaytı çok iyi anlatmanın faydası çok büyüktür. Yani sunumlarda ilk başı ve sonu çok vurucu yaparsan hakikaten ortasına göre çok daha fazla kredi verirler sana. Aynı şekilde makale yazarken de mesela son figüre, yani sırayı bozup son figüre en iyi figür yapmaya çalışır. En iyi figürleri sona koymaya çalışırlar benzer bir şekilde. İnsanın aklında çünkü hep en sondaki kalır. O da çok iyi oldu yani bunun son slide olması. Bu kadar benim yorumlarım. Bu vardı bir de, doğru. Bu da güzel oldu. Belki ilk claim için bir tane slide yapmışsın. İkinci claim için ayrı bir slide yapmak iyi olabilirdi.
Evet.
Ben bu senin sunumunu ve örnek sunum olarak almak istiyorum. Ve bizim bu tartışmamızı benim şansına verdiğim feedbackleri ki zaman ayırıp çalışmak isteyen olursa hani neye benzetmesi gerektiğini sunumu görsün. Senin için bir sakıncası var mı?
Yok.
Tamam.
Bana da gönderebiliyor musunuz peki? Yoksa bana mı?
Videoyu görmek mi istiyorsun?
Evet.
Tabii onu koyacağım yerde mutlaka görebileceğim bir yer olacak.
Tamam.
Çok teşekkür ediyorum tekrar Eylül. Seninle
Ben
gurur
teşekkür
duyuyorum. Yaptığın sunumla daha doğrusu gurur duyuyorum. Her
Teşekkür
zaman bu
ederim.
kadar iyi yapmak zorunda değilsin yani baskı gibi olmasın. Ama bu yaptığın iş gerçekten alkışı hak ediyor.
Teşekkür ederim hocam.
Görüşmek üzere. Hoşça kal.
Görüşürüz.